Can you say something?
1, 2.
Maybe it's too loud.
It's too loud.
I think so. It's 8. Is it 8? Can you see? It's 8, yeah.
1, 2. Yeah, it's 8.
Can you say something now?
1, 2.
Better.
Is it better, or?
Yeah.
Okay. And
Eins, two.
I just put you on stage?
However you want, honestly. If it's easier for you, I'm on stage, that's fine. It's a quick announcement. It's going to be like 2 minutes or something.
That's good. Yeah, then I just move it.
Thank you. I'll be behind you maybe. Maybe I said like in this room just for the time being.
Go-to man. I like it. I like it. It's important to know people like this. People who can't say no. No. You're too late now, Max. You can't say no now. I'm not going to help you.
Just chiefly to the speaker slot. It's no problem. I will talk about.
I'll do that. I'll do that.
I have something ready.
You put your resignation.
You have the. This is the right one. Especially with this chat teaser. They asked me yesterday. All right. All right. Let's let's start on time. Can you close the doors, please? Thank you. Okay, let's start on time. We have a very interesting packed schedule about AI guidelines and 2 talks about T cells. So we are here not to collect praise, but get valuable feedback for our projects. There are some announcements to make. Is there any new CeMM members today?
Please welcome with me Agnieszka Kraft, who joined us directly from ETH Zurich, the computational side of the Bock Lab.
Today there is also SCP-1 right after this. There will be next week the SEM Christmas family event and Christmas party. And Bernardo also has some announcements to make for this.
So thank you, Max. So as you know, next week will be the Christmas party, so we're all very excited to bring it to you. The people that are registered should have already received an email confirming. If you, for some reason, can't attend the event, I would ask that please follow the link that you got in the email and cancel. And we would also like to ask you, please, to show up on time at 5:30, so that then we can organize everyone and then follow up for the tours and for our events, keeping everything in schedule. Also, would you mind switching to the next slide, please? So, as you all know, there's also going to be a charity associated with this event. So we will be donating in favor of the children that are affected by the war in Ukraine. And Giulio has very gracefully offered for every euro that we donate, he will also donate €1. So please, everyone, pitch in so that we can try to bankrupt him as much as possible. For every €4 that you donate, you'll also get a ticket for a raffle, which has very cool prizes. And for every €10, you'll get 3 tickets. So the more you spend, the more chances you have of also winning these cool prizes. So thank you all very much, and we hope to see you then at the event.
And with this, I would like to start the first talk by Micha and Ulrich about AI guidelines.
Great, thank you. So welcome everyone, big audience today. So we will do an AI literacy training today, and the goal is to help all of you use AI in a responsible, transparent, ethical and secure way. So it should help you, but it should also help CeMM to comply with European regulations on this topic. So today I will start with presenting briefly the AI policy that we drafted for CeMM. Then Misha will introduce available AI tools here at CeMM. And then in the end, if there is time left, we will quickly go through a couple of usage scenarios. So I will just start with the AI policy. So it's a document available on the CeMM intranet. It covers different guidelines and regulations for using AI systems. And this actually applies to all kinds of AI systems. So it could be third-party systems where you have an account. It could be the AI systems that you develop yourself for your research. It could be generative AI or predictive AI, image analysis, and so on. So all AI systems are covered, should be covered here. And basically, there are 5 guiding principles in this policy, and I will now go through those 5. So the first important aspect is that you are responsible. So you're the pilot, not the passenger here. You're responsible for any work that you produce using an AI system. And this is how it typically looks like. You have some tasks that you formulate. This is what's called the prompt. Then you provide some additional information, maybe, which is then called the context. You feed this into an AI system, and it will produce an output. This could be some generated text, could also be a prediction or whatever. so you're responsible for whatever you produce here. And this also means that you have to be aware that the AI systems have, of course, limitations and also training biases. So they can generate inaccurate output. They can also fabricate content, which is called hallucination. To best use them, you have to provide very clear and precise input. And you should always very critically assess and also verify the output. So what are those biases? I have a very brief and simple example. So just 2 days ago, I put this into the ChatGPT 5.1, translated this sentence to German. The doctor hired a secretary. And if you translate it to German, it will pick the male version of the doctor and the female version of the secretary. And this is still happening today. It's not like in the early days of the LLMs. Now, more interesting, if I add some adjectives to the secretary, assertive and decisive, then it will be the neutral version, not the female anymore. This is a very obvious thing of a bias, and this is already discussed in the literature for years. There you can easily spot it. You have to be aware of it. But of course, any other AI system with much more complex tasks than just translating will also have the training biases, where it's much harder to identify those. So just be aware, keep in mind that the training data will always affect the output. And also think about academic integrity and the ethical standards that we have. So you should not use those tools to replace your own thinking. You should contribute your own ideas and also disclose the usage of AI. More on this later. The second point is that you should evaluate your use case. So you should do a risk classification. This is coming from the EU regulations. So they define 4 different risk categories for the use of AI. It starts with minimal risk, which is basically just minor tasks. The implications are, there are no implications of using AI for a minimal risk use case. I would still recommend to be transparent about it. And examples are using it for grammar correction, for speech recognition, or for formatting text. The second level is limited risk, so there content is generated or manipulated. Here, it is required to be transparent, and the generated content should also be labeled. Examples here is a writing or coding assistant, or summarizing literature, or modifying images, or whatever. So this will be probably most of the use cases that you typically have. Then there are also high-risk use cases. So this is when the use of AI affects some sensitive decision. So it could be about health, about safety, about employment, education, or otherwise touching fundamental rights of individuals. The implications are then not only to be transparent, but also there is very strict regulations on how to document the use or the system itself if you develop one. There has to be made sure that there is human oversight involved, so no automatic decision should be taken. It's always possible for a human to intervene or to check on the results. And in some cases, you will also have to do this so-called fundamental rights impact assessment. If you develop if you not only use such a high-risk system in a high-risk use case, but also develop such a high-risk system, then there is also a database where you have to register your system, your AI system within the EU. And examples here are performance evaluation of employees or grading students based on their work or their performance, using AI systems for diagnostic or treatment decisions, or also biometric profiling. And then there is another category which is unacceptable risk, which is basically just prohibited. So it's against the law to use those in those cases. And there are not a lot of cases, I think, where it would affect us. but the examples that I found are social scoring of employees or emotion inference, interestingly, is also one of these prohibited use of AI. This is a very simplified overview. There's the regulation are much more detailed, but this is like it should help you to identify the risk. So if you do like if you have students in a classroom and you film them, and based on vision, computational vision, you try to infer their emotions while you're doing your lecture. So you want to get like not only the feedback that they tell you, but also the feedback that they show with their visual expression or whatever. And if you process this using AI, then this is not allowed. It's not emotion inference is not prohibited in general, but in certain settings like education or employment. An important aspect, if you should always also document your risk classification. So if you have some tasks, AI tasks, you should actually document that you did this risk classification and how you came to this decision. The next point is to actually choose the right tool. So we have also on the internet an AI inventory where we list all the available tools. Misha will talk about them in a second. Try to choose the system that fits your task. For trivial tasks, you use a standard system. For advanced tasks, you use some specialized tools. Or for complex workloads, you might need an agentic AI system. Use the systems for their intended purpose. So don't misuse them. Also consider the impact on resources and environment. So resources, it's of course also monetary, all of these costs. But also it uses up a lot of energy, so also be aware of this. If you develop AI systems in-house, it will also take up our cluster resources, so also be aware of that. If you are developing systems, please also register them in this inventory. Let us know so we can add them. Another point that should help you to choose the right tool is that you should also respect privacy and IP. Here it's very important to say that the systems that are behind all these free or personal accounts, they in general do not comply with data protection standards that we should adhere to, for example, the GDPR. So they might store the data, reuse them, and maybe even share them with third parties. So don't use those systems for your work. Instead, use the systems that Misha will present. And also, important, evaluate the use case on data sensitivity. If you're working with very highly sensitive data, use on-premise systems. So they are running in-house. We don't have this yet. This is work in progress. If you use AI with sensitive or identifiable data, use the systems that are in on-premise or in the cloud environment, so the Microsoft systems. If you use non-sensitive data, then you can use these third-party systems that we have with enterprise contracts. Also, always think about the data privacy, GDPR rules, respect the licensing and the usage restrictions of the AI-generated content. So you cannot do everything with the AI-generated content. There are, depending on the provider, also usage restrictions. And if you are developing an AI tool, respect, of course, also the licensing terms of training data. And the last point, the fifth point, is to ensure the transparency and research integrity. Document your usage, I already said, document the risk assessment, but also document in general usage. Keep track of the input and outputs. I know this sounds very much work, but for very simple cases, you probably don't need it, but for high-risk use cases, for sure you need it. And always disclose when AI contributes to your research or analysis or decision-making. So one example that looks like this. You just add in the end, this document was prepared with assistance from Microsoft Copilot for information retrieval and copy editing, for example. There is a small exception, this editorial control exception. If it's a small task and you fully review and edit the output and hold the editorial responsibility, then disclosure may not be required. But in general, it's always recommended to disclose the use. For developers, there's another thing on ensuring transparency. So you have to inform your users about the AI use in your tool, and you have to label all the output that it generates in a machine-readable format. That labeled that this was AI generated. Okay, this was a very quick tour on the policy. Now we'll hand over to Misha to present the AI tools.
Yeah, thank you, Ulrich. He did well in explaining all of this, and now I'm in charge of screwing up the live demo. So basically, these are just screenshots. Right now we have two tools. There's Microsoft Copilot, which is available. This is a bit quiet, which is available via the browser. You see the link here, but you also see all the links and all the information you see here in this presentation is also in the intranet. And the other tool is this CeMM chat based on OpenWeb UI. And I try to go live to the website now and show you two small use cases, what we presented or prepared. This is basically how to write a polite email, and this is kind of something what I use daily to just... My emails are normally not polite, but yeah, I try to be more polite. And AI helps me with this, and basically you can just put it into Copilot and ask him to write a proper version of an email which you can use in a professional environment. And
have to scroll down.
And that's basically it. So very polite, very nice. This is how I roll these days. The second live task. So there's this OpenWeb UI. You can go to with your favorite browser to chat.cem.at and sign up for a login. There are all the tools we have you have available right now. You can go to this drop-down list. You can choose every default model you want to use. Basically, with the exclamation mark, there is also a small description, what is it good for, how much it costs, and how you should use it. The key message is you should not use deep thinking models for daily tasks, yeah, because it's just burning money and it's not very useful. But you should think about the proper use of the tools.
To the Opus again.
And it switched to the yes. For example, if I would, so this use case is now we uploaded 2 documents, and we want to combine them, and the tool should yes. The tool should help us to write the grant proposal, and with Gemini this would be a nightmare because it's not built for this. But we use the Opus. Claude Opus, for example, is a deep thinking model which can do that. Very nice. And very important, if you upload a document, and if you don't say use the entire document, AI will already use a summary of this document, and the output is not that great. This is, you have to keep it in mind, and then it's using the full documents. And then the AI model tries to give us a good output, the combination of these 2 models, and within seconds you have a very useful first draft of a grant proposal.
Thank you, Misha.
I almost didn't screw it up. Yeah. It continues.
This is
The deep thinking models are quite slow, but this is for a good reason, because the output is so, they think a lot about it and the output is quite nice. We have a good overview on the intranet, which tool to use when, and is also a work in progress, so we will add constantly more models to the systems.
Is there a limit on it, how much we can use it?
Thank you. Yes, if you use one of the Claude models for scripting and coding, you are burned through millions of tokens quite easily. Yeah? Right now, there's CeMM is paying for the amount of tokens you use. Yeah, it's connected directly to the API models, and we are just monitor it. And if we use too much, then we have to think about a better strategy. It really depends what the user base is and what you use it for. Yeah. But it's not for free, I can tell you that. So with Claude, you easily can spend 200 euros per day almost.
Maybe you could make a guideline where it says more specifically which model you could use for which complex.
It's already in the intranet.
We started something on the intranet. If you think this is, you want more detail on certain aspect, let us know.
All right, in the sake of time, please.
Yes, we will finish. So as you could see, this is very impressive. It will generate even the Gantt chart and tables and a 12-page research proposal. Of course, talking about academic integrity, you will not use this and submit it to FWF. This is for you to get an inspiration. It's not. It should not replace your work. It should help you. So we did the live demonstration, and then I also have prepared some usage scenarios which we will skip for today, but you can also look up the slides later on. They will be also on the internet. It's basically just getting you to think about which system to use, which use case you have, and so on. So yeah, that's it. In the end, there are the take-home message you already know, and if there are any questions, let us know. Check the intranet. Write to ai.cem@cem.at if you need something. If it's about privacy, ask Misha. If it's about IP, ask Pedro. And anything else, you can ask the both of us. Thank you. Very brief announcement. Still, you need to sign that you were here because this is a mandatory thing. So before you go, make sure you put your signature here. Thank you.
Can you distribute the slides?
Yes.
All right, for the sake of time, we immediately go to the next talk, which is the first talk about T cells in the skin.
Yeah, thank you, Felix, for the Max, sorry, for the introduction. I'm Laura. I'm a postdoc in the Stari Lab, and today I will once again talk about my PhD project, hopefully for the last time, but it's about tissue-resident memory T-cell subsets in the skin, and we looked at this in the context of allogeneic hematopoietic stem cell transplantation, and we found that the balance between different subsets is disrupted in these patients. Since my last Friday seminar, I defended my thesis and we submitted the manuscript and are currently working on revisions. I also contributed to the WWTF precision medicine proposal that Georg handed in, and I completed this fast-forward project management class that was offered by Sam. So back to the project. As I already told you, we are interested in tissue-resident memory T cells, and these are a subset of memory T cells that permanently reside in peripheral tissues, such as the skin. And they are very important in protecting the tissue from secondary encounters with an antigen and mediating rapid recall responses. And we look at this in the context of stem cell transplantation because we observed in previous studies that these cells in the tissues can survive the myeloablative conditioning that these patients receive in order to deplete the circulating immune cells and the cancer blasts. And when these patients then receive the stem cell transplantation, new donor TRM are formed in the tissue next to this host TRM that survive, and there is a mixed chimerism in these tissues for decades after transplantation. And these patients that receive this allogeneic transplants are also at a very high risk to develop graft-versus-host disease, and this often manifests in the skin. And it's basically an inflammatory condition that is first induced during this conditioning phase where tissue injury occurs, that then triggers activation of antigen-presenting cells that prime allogeneic T-cell responses in secondary lymphoid organs, and these then infiltrate back to the target tissues and mediate tissue damage there. And we want to look in these patients both in homeostatic conditions, that is when the immune system is repopulating in the absence of inflammation, but also in GVHD in this inflammatory context, how this affects the TRM populations in the tissue, and also how it affects the ratio between the host and donor cells, and if we can learn something about the dynamics of these different subsets by looking into this host-donor cell dynamics and learn something about human TRM biology in a kind of in vivo system, because most of what we know about TRM biology comes from mouse studies where you can do adoptive transfer, parabiotics experiments, and so on, and this we cannot so easily do in humans. And we perform single-cell sequencing for this project, but I already talked about the analysis of this data set in my previous presentation, so today I just want to briefly remind you that this is the cohort that we sampled where we looked before transplantation, then on the day of transplantation, and 14 and 100 days after in the first cohort, where we took the serial samples, both blood and skin. And then we also had a second cohort of patients that developed acute cutaneous graft-versus-host disease, and we collected untreated skin lesions from these patients, and blood as well. And very briefly, what were our main findings when we looked into the T cells? We found this gene expression profile that was upregulated at baseline, downregulated in GVHD, which very closely mapped to this distinction between these two TRM subsets that at this time were described in mice, where they basically showed for the first time in this paper that there are these two different TRM subsets that have different maintenance and development mechanisms. So before it was always thought that all TRM cells depend on this Hobbit transcription factor, but this was the first paper that showed in mice that there is a second TRM subset that can establish residency with this CeMM transcription factor pathway, and that also that these cells can specifically affect it when you block, for example, signaling through the IL-7 receptor, and if you knock out this CeMM transcription factor. And we then also looked for this cluster in the CD8 TRM compartment in the skin, and we found that the phenotype of these cells matches these mouse TRM17 cells, and this cluster is also reduced after transplantation. And when we look into the host-donor dynamics, you can see that it seems that both the host cells cannot maintain this lineage and the donor cells do not repopulate it, whereas in the cells that are not in this TRM17 cluster, this looks different. So this suggests that there are different TRM populations in the skin that have also in the human system probably different requirements for their maintenance and their survival. But this was just like based on our relatively small single-cell cohort, so we also wanted to validate this on protein level. And we also had this problem that we only have the day 14 and day 100 time points after transplant, so we couldn't really say if this is an effect that is specific to the GVHD. It seems to be still here on day 100, but we have very few patients, so we also wanted to have an additional control cohort. So we tried to sample patients that at the same time point as our GVHD patients, they're at the same time point after transplantation, but they don't at this time have an ongoing GVHD, which is logistically difficult, but we still managed to recruit some patients, and this was actually quite interesting because we could see that actually this downregulation of these TRM17 markers seems to already be there even if the patients don't have GVHD. So it's not really an effect of the inflammation, but rather of the transplant regimen itself. And this was even more surprising that also these type 1 cytokines, interferon gamma, and this marker for the TRM1 lineage, CD49A, is also upregulated even if there is no GVHD after stimulation in this case, but this suggests that the skin after transplant is already primed for these type 1 immune responses that then would characterize a GVHD lesion. and that in some susceptible individuals, this might then lead to the development of a GVHD for whatever reason. Then we got asked by the reviewers if we can do this validation analysis also separately for host and donor cells, which is not so trivial because we cannot just easily sample new skin biopsies and look at the chimerism in a time frame that is compatible with a revision. So what we proposed now is that we will make use of these circulating T cells with resident phenotype that we described previously, and that also have been described in mice, which is basically a new concept that says that cells that already were TRM, fully differentiated TRM, can redifferentiate and go back to the bloodstream, but they keep their TRM phenotype at least to a certain extent. And in this previous publication, we could show that these cells in the blood, even though it's a very small population among the memory cells, they mirror the mixed chimerism that we have in the skin TRM, whereas the other blood cells do not, because they're mostly donor cells. And so we were wondering if we can use these cells now to also look into this distinction between the TRM17 polarized cells among this CTRM pool. So what we first needed to confirm is that this CTRM actually also mirror the heterogeneity between TRM17 and the other subsets that we see in healthy skin. So when we look at this and gate for the markers that we before saw were differentially expressed in the GVHD lesions, at least in healthy skin, it seems that this blood-derived CTRM have exactly the same phenotype as the TRM counterparts in the skin. So they seem to really mirror the phenotype of the TRM in the skin in the healthy setting. And then when we look into the same cohorts that we already looked into in the skin, they also have the same expression patterns as the TRM counterparts in the skin. So also they mirror the phenotypic changes that we observe in the patients after transplantation. So this made us relatively confident with that we can also use these cells from the blood now to look into the chimerism. And we again plan to do this now with the XY fish, so fish staining for the sex chromosomes, so that we can then identify which cell in either this CTRM17 gate or in the remaining cells are host or donor derived. And this is what I'm currently doing. I'm compiling these cohorts, trying to, together with our MDs in the lab, recruit the patients that had the sex mismatch transplantation in these different time points after transplantation, and then we will hopefully in the next weeks do these XY fish stainings. And I just did a first check to see if the protocol still works in my hands, and this is then what it would look like, where you then have these fish spots in the nuclei. Then, since the last Friday seminar, we were also asked to do a cell-cell communication analysis from cells that are in the tissue microenvironment that might drive this type 1 shift. So we first looked into the other cells in the skin and which ones of those have the highest differentially expressed genes compared to baseline, and these were fibroblasts, macrophages, and dendritic cells. And you can already see that among the top upregulated genes in all three subsets are chemokines like CXCL9, CXCL10, CXCL11 that will preferentially recruit CXCR3 expressing T cells, which are usually the type 1 polarized ones. And we then put this into a cell-cell communication tool like CellChat. And when you then get these communication plots, you can see here that at baseline we have signaling through CCR6 and CXCR3 and also IL-7 and IL-15 that would basically correspond to either this TRM1 subset or the TRM17 subset. And when we then do the same plot in the GVHD, this CXCR3 signaling is relatively expanded and the IL-7 signaling is relatively decreased, indicating that there could be a trend that also the microenvironment favors more the establishment and the recruitment of type 1 polarized cells over these type 17 lineage factors that would be trophic for the TRM17. And we were also asked to validate this in situ. So right now I was establishing IF staining panels so that we can assess the protein levels of these host ligand and receptors in the skin. And this is the first panel that we plan to stain for, where we stain for IL-15 and then CD215, which is the receptor that is cross-presenting IL-15 to T cells. And at least on this image, it seems that there are more cells in the dermis that express both IL-15 and the presenting receptor, but this I still need to analyze. And then the second panel would be for the CXCL9 and CXCR3 axis. This also, at least the staining seems to work. And for the third part, where we want to look into IL-7 signaling, I'm still working on establishing this panel, but hopefully in the next weeks and after Christmas, this should all be finished, and I hope it will be significant. Let's see. And then lastly, what we also wanted to assess if whether this is a permanent effect or if eventually the homeostasis between the different subsets, or at least the markers that were differentially regulated, goes back to normal. So we made use of this cohort where we had skin biopsies embedded in OCT already from patients that were at least 1 year after transplantation and had like clinically recovered, meaning that they weren't taking immunosuppressants anymore and otherwise didn't have any further complications. And here it seems that this CD127 expression normalized, it goes back to the levels that we see in healthy skin. And also there is no difference whether these patients that are 1 year after transplantation before had a GVHD or not. And then the same is true when we look into this ICOS expression. And this was actually also quite surprising because in mice it was described that ICOS is a specific marker for the TRM17 cells. And when you target ICOS, you can specifically deplete these cells. But in humans, it really doesn't seem to be the case that this ICOS is specific for the TRM17. It's rather only upregulated in the GVHD, so more of a context-dependent upregulation rather on the type 1 cells. So this, I think, is also important to know that this in humans might be very different. And this is already the end of my presentation. So we are currently proposing that there is this decreased survival of these hosts, TRM17, and also a delayed reconstitution of these cells. And this in GVHD lesions might be even more amplified by this signaling that favors the type 1 polarized cells. And this might also have implications, of course, if we can find out how to specifically target these TRM17 cells over TRM1 cells in other skin diseases as well, where, for example, TRM17 cells are the main mediators of disease like psoriasis or others like that. And regarding feedback, if you can think of any additional experiments that we could still do before we resubmit the paper and anything that could improve the story, please let me know. That's it.
Thank you for this very, very inspiring translational study. I see many input. Unfortunately, due to the sake of time, I saw you first, so you can give one comment.
Thanks, very nice. I was wondering whether relying on the sex mismatched donor-recipient pairs could also introduce some bias, because there's all sorts of sex-specific differences, especially in the immune system that we know and some that we don't.
Yes.
So have you tried to genotype the single cells you have originally from the data you have? Because it will be extremely sparse, but all you need to do is to basically see all the SNPs across all the genes and aggregate them per cell to know which donor they come from.
For the single cell data set, we did exactly this to annotate the host donor genotype, but then for like experimental validation, it's just a bit easier to go with the sex mismatch, because otherwise for the in situ imaging, for example, we would need to stain for these for lesions basically or for SNPs in the genome, which is not so easy. So it's easier experimentally to do it with the sex mismatch, but it would be, of course, nice if we could do it based on the SNPs, and it also would allow us to recruit more patients, because not so many do get a sex mismatch transplants, exactly for this reason that it's a bit higher risk to develop GVHD. Yeah.
I'm sure in the break, at the breakfast, Clarissa and Chris can give you also more input. And from the T cells in the skin, we now travel to the T cells in the gut. Chris.
Thanks, and good morning from my side as well. I'm Chris. I'm a postdoc at Clarissa's lab, and today I will talk to you about neuronal and immune cell interaction. So we all know that neurons feed every organ on your body. So over the last few years, it has been established that actually neurons can also regulate and influence very hardly your immune response. So one of the original or the pioneer studies relied and described this so-called anti-inflammatory reflex. So this was originally described happening in the spleen, where norepinephrine-producing neurons infiltrate the organ, and then this activation and production of norepinephrine by those neurons leads to a differentiation of a specific type of T cell that actually expresses the rate-limiting enzyme for the production of acetylcholine, which is called CHAT. These T cells produce then acetylcholine and then act on other immune cells as macrophages to dampen their pro-inflammatory immune response, like IL-6, TNF-alpha. So this was found very important to regulate systemic inflammation, but the same can be thought or has been partially shown to happen in the gut, where you also have this separation of tissues where on the outside of the gut you have the muscular layer or the myenteric layer where it's highly infiltrated with cholinergic neurons, while norepinephrine-producing neurons can go more into the lamina propria and even the villi. And there in the gut, you also find some CHAT-expressing T cells, and you can also think that the acetylcholine production by these T cells can influence immune responses in situ, so in the gut. So we know that the gut is like a home for a multitude of antigens. You have to deal with frequent or always present bacterial antigens, but then you also have to deal with food-derived antigens. And this follows kind of a density gradient where in the small intestine you have higher abundance of food-derived antigens, while you don't have as many bacteria in the proximal side, but then this starts increasing, reaching into the distal side, where you have a nice blurry or mixture of food antigens and bacteria antigens. Where then on the colon and large intestine, you have high amounts of bacteria antigens, not so many more food-derived antigens. So the body, we know that it has to deal with all of this input and try at homeostasis not to get overwhelmed and have constant inflammation. So we thought about this very much in the lab, and Clarissa, before I arrived, has established this mouse model where we want to investigate how the system, how the host responds and deals with these challenges. So we thought about this model, and please pay attention because it will be the model for the entirety of the presentation, where we fast all the mice by a certain time, so 9 hours, and then we divide the groups of the mice, and then we refeed half of them, and the others we kept them starved. Like this, we can answer how the body and how the system actually responds to feeding and to challenges that increase the antigen load and bacteria metabolism. And this has been shown also before in our lab, that upon this refeed, you have increased bacterial density, but also bacteria are way more active, metabolically active. And I've also shown that at the time point, so 5 hours after, your T cells are actually slightly responding to this input. So you have increased production of cytokines like IL-17, but also importantly IL-10. So we think that this is a response that the body needs to have frequently, that after this challenge, you need to dampen and respond to the insult. So we thought, okay, we know that acetylcholine is important for gut motility, and also it regulates inflammation. So then we thought, okay, maybe acetylcholine also can play a role in this model. So we went into a drastic system where we before the refeed, we treated the mice either with muscarinic acetylcholine receptor inhibitors or nicotinic acetylcholine receptor inhibitors. These are FDA-approved drugs. And then we look generally big immunophenotyping, and we start seeing that the mice that actually were treated and refed with nicotinic receptor inhibitors showed some differences on immune response. So we are still slightly underpowered, but you already see some differences, meaning that in the refeed setting with the nicotinic blockage, you don't have this IL-17 increase. And you, on the other hand, in your innate lymphoid cells, you start seeing pro-inflammatory cytokines being produced, like IL-22. And you look at your myeloid cells, and you see, so normally, under refeed circumstances, 5 hours after refeed, your CDC2, so conventional dendritic cell 2s, seem to be less mature. They downregulate CD86. However, when we block the nicotinic, this goes back to the normal, and we start seeing some neutrophils coming in. So acetylcholine signaling in general seems to be regulating homeostasis response against feeding. So then we thought, okay, we know that immune cells express this acetylcholine, can produce acetylcholine. So we got these chat GFP mice that report expression of the rate-limiting enzyme for acetylcholine production, and we looked at the immune cells in different tissues. So it has been reported also that B cells can produce acetylcholine, and they are highly prevalent in the secondary lymphoid organs like lymph nodes, Peyer's patches, or spleens. However, when you look into the gut itself, so proximal, distal, small intestine, and large intestine, you start seeing that T cells and also ILCs start being the main producers of acetylcholine. Now, what about identity? Are these acetylcholine-producing T cells kind of prominent in a specific lineage of T cells? So we also sorted these cells, stained for transcription factors, and so ROGAMA T is the transcription factor key for Th17, FOXP3 for Tregs, and then you have these double positive cells, which are known as the peripherally induced Tregs. They are the ones known to be specific against bacteria or food antigens. And we see that in the chat positive T cells, you see a highly enrichment for this Th17. It has also been reported, but also importantly, there's also a frequency of these P-Tregs that also increased in the chat positive T cells. And this is most notably in the large intestine, where you also have higher amounts of bacteria. So then we thought, okay, the body, the intestine, has to deal with all of these antigens, as I mentioned before. So we kind of wanted to model this into our system. And we used two systems to model antigens. So for food-derived antigen, we have ovalbumin that the mice never saw. So then we can induce a different antigen to the food. And then we also have a commensal antigen, a commensal bacteria that our mice have, which is segmented filamentous bacteria. And this is known to induce Th17 T cells in the gut. So then we also invented kind of two systems to check how chat-positive T cells can be induced by either of those antigens. So first, we generated mice that harbor a TCR transgene specific against ovalbumin and also harbor our construct for chat-GFP. We transferred these T cells. We gave the challenge, so ovalbumin. We waited a few days so that the antigen is washed out. And then we do our fast refeed experiment, having either the ovalbumin or having a mock antigen, so antigen that is not specific for those T cells. And we start seeing that, yes, chat induction seems to be antigen specific, and food-derived antigens can induce chat expression. Then, the other way around, we wanted to find out, okay, how these chat-positive T cells are mediated by the microbiome. So we generated, again, transgenic mice harboring the TCR receptor that is specific against this bacteria, SFB, also harboring the chat construct. And we did the same experiment. We transferred the T cells. We didn't need to give the antigen because the mouse already has the antigen as a commensal. And then we did the fast refeed strategy. And we saw that a lot of these transferred cells express high levels of chat. So yes, chat can also be induced by microbial antigens. So with this data, we thought, okay, then it's like a T cell or antigen-driven stimulation. And to validate also the microbial-derived antigen, we just treated the mice with a mixture of antibiotics for two different time points, so 1 week or 3 weeks. And we saw that actually in long-term antibiotics, chat levels are also going down on the T cells. So antigen availability is actually a key input for these T cells to produce acetylcholine. So yeah, so we have this. However, it's still an artificial system with transplanted transgenic T cells. So we wanted to start looking into the physiological T cells that already exist in the mouse without putting foreigner T cells in there. And this we could do by using tetramers. So we designed tetramers in which that basically have the sequence of the segmented filamentous bacteria, and we can identify physiologically T cells that have TCR that recognizes this SFB. And we also generated these mice that have a double reporter, so it has the chat construct, but also report ROGAMA T expression, so it means that we can also find if they are Th17 driven. Sorry about the quality, I don't know it always happens with this plot, but basically we find a few cells, so this is like around 1% of the CD4 T cells, and then we can divide chat positive versus chat negative T cells. So we see that these physiological T cells that recognize antigen can also express chat. And we use this system importantly because we wanted to find out what makes these cells special. How different are the T cells that recognize the same antigen, but half of them have chat and the other one doesn't have chat. So we use this to sort these cells out and perform small low-input RNA-seq. We found a few genes that are differentially expressed, and I will highlight a few of them. So ID2 transcription factor might be important for regulation. IL-23 receptor might be one of the signals that these T cells need. And we also saw some increased IL-10 expression by the chat-expressing T cells, so meaning that although these cells are Th17, they also can produce IL-10, and specifically the cholinergic producing cells. And out of the blue, we also saw some weird genes that are actually expressed or functional active in neurons. So this will make sense that these T cells somehow have a signature or have some weird genes that neurons will express, although it has not been reported in literature. At least nobody knows very well what these genes do. So we are also very curious to see if we can find some functionality for these genes. So yeah, highlighting again this IL-10, we found it curious. So we see that this IL-10 is also upregulated in our feeding condition, and for some reason, acetylcholine also regulates the feeding response. So we thought, okay, let's see if these T cells are also the ones responding and producing IL-10 in our feeding system. So yeah, we did our fast refeed system. In our chat GFP mice, we find chat-expressing T cells. They are not differently abundant, so they are there in the fasted condition. They are there in the refeed condition. But we then looked into the IL-10, which is one of these markers. We also look at IL-17. And we started seeing that, yes, these chat-expressing T cells, not only at the fasting level, so if you compare the blacks, these chat-expressing T cells are preferentially expressing IL-10, but then when you refeed, these are the ones that are contributing to that IL-10 pool that is increased. So these are the fast responders, and those are the cells giving the IL-10, so the anti-inflammatory signal to the tissue. And then finally, we were also curious, are those cells actually the food antigen specific, or are those actually the ones responding to the microbial increase that we saw originally? So we went again to our system, transplanted these SFB specific T cells. So we used this as a proxy for the commensal bacteria increase. We transferred these cells, did the fast refeed, and voila, we see that actually only on the refeed setting, the microbial specific T cells are producing IL-10. So not only the refeed induces the response, but then the response is not only driven by the food antigen, but also by the microbial input. So yes, so it's basically what I said. I told you microspecific T cells are also responsive to the feeding status. So giving all of this data, our next steps is based on our RNA-seq, and we are also performing a tag-seq from this chat versus non-chat expressings, is to then find possible regulators of these chat-expressing T cells, and then use our system of transgene transfers to actually validate and find regulators of chat expression, but then maybe also IL-10 response against the antigen. So as an overall, so a summary, if you eat enough pizza or apples, you have then a lot of these antigens in your gut, and then these antigens can be translocated into the tissue. They can be normally, can migrate or can be transferred by antigen-presenting cells into the lymph nodes, and then, or also can be presented locally, and then these T cells that recognize these antigens will also not only produce acetylcholine, but then they will also produce IL-10. And these signals, we think, are important to act on another cells and more in the innate system side and kind of regulate their expression and their pro-inflammatory response. So we think this is a pathway that it's kind of needed to keep the homeostatic response against your normal frequent diary or like diet and daily responses. So then, of course, the key thing that it's missing is to actually prove that these differences caused by acetylcholine are actually derived from the T cells. So we already have these mice that are depleted of this enzyme, specifically on the T cells or in certain populations of T cells. So these are coming in the next weeks, the experiments. Then we also generated and received already a big single-cell data set of these fast and refit conditions, where we took special care to have a lot of these chat-expressing T cells so that we can also analyze them. And we are also going into inflammatory models. So far, we have this model of enteritis. So we administer anti-CD3 antibody, which causes inflammation mainly on the distal small intestine, but also in the proximal small intestine. And we want to see there if acetylcholine is also important in the T-cell mediated inflammation. And giving also the importance of IL-10 for tumor immunity, where I'm also thinking in long term to perhaps follow a bit into the tumor and colorectal cancer driven, because it's not really known if cholinergic signaling actually plays a big role in the anti-tumor immunity. So as overall, I showed you today the chat expression is not linked to a particular T-cell lineage phenotype. Microbial and food antigens can trigger and differentiate these cells. Chat-expressing T cells are providing anti-inflammatory signals in the tissue, and that the acetylcholine is important for this postprandial response and keeping the homeostasis in the gut. So feedback required, I want to analyze a little bit of localization in the gut. So I want to see association of these T cells if they are in close proximity with neurons that are infiltrating the villi and in the gut. So I'm not a big immunofluorescence guy. I haven't done many, but we are starting to go in there. So if you have any tools or tips for stainings, please come and chat with me. And yeah, thanks a lot, the lab.
Thanks a lot for this very impressive presentation,
Unbiased.
especially since you just came back 3 days from your wedding cruise.
Thank you.
We have time for one question, maybe.
Hi. Thank you. Very interesting talk.
Thanks.
So I was wondering, the acetylcholine itself is also a very important neurotransmitter that is involved in arousal and attention, and also specifically in the feeding and satiety circuitry.
Yes.
Do you think there is any kind of feedback mechanism from the peripheral nervous system in the gut, maybe to the CNS back during this process that you described?
Yes, it's a great question. So it has been shown that acetylcholine goes up, especially also in the hypothalamus, 30 minutes after you have a meal. And this could be linked to the appetite. And actually what we saw is that in the blockage experiment with the nicotinic blockage, which we gave it gavage, so it should act first locally, but it eventually, I guess, gets systemic, is that these mice tend to eat half of the amount of the other mice. So this is a confounding factor of that data. And it could also be other factor is that the gut motility, right? Acetylcholine has also been shown to be important for the gut motility. And yeah, so these are confounding, and I do think it could play a role. The question is, are the T cells being the ones giving that signal? I doubt, but it will be a striking finding. But we have the T cell knockouts, which what I'm curious to look. And I will have these measurements, yes. Yeah, thanks.
Then maybe time for one really quick comment I saw over there.
Yeah, this gets more and more fascinating every time. So how you mentioned the involvement of innate immunity. Do you think that with the food you will also have xenobiotes, small molecules that will trigger that is required for your antigen-driven responses to the?
Yeah.
Is there a way we can study this? Because maybe it can be studied in some sort of in vitro system where I can systematically see what the food components would be that would trigger or help the response?
Yes, great question. So we do have data generated previous from the lab where we did this fast refeed in the presence or absence of antibiotics, for example, and did metabolomics on those. And there are a lot of metabolites that are differentially expressed, produced, and that are actually dependent on the presence of microbiome. So this we are investigating. And the other part of the question, so the food-derived components. this we are also starting to investigate. We will do these fast refeed experiments with enriched diets for lipids, sugars, and things, and other things, or high protein or low fiber, or this type of things, and we will investigate that. So we are starting to go into that direction, mainly also with Dina, a PhD in the lab, we are going into there. Yeah.
Thanks a lot for this really packed Friday seminar.
Thanks.
And maybe give a round of applause for the speakers today.